Thank you very much, Zan, for agreeing to give a Build a Cell seminar, and you can get started.
Yeah, well, good morning, everybody. Yeah, I had the pleasure of meeting Kate one-on-one at a meeting of Build a Cell that took place in China. So I have the T-shirt on from that meeting. And I'm going to tell you like what we've done with trying to develop 4D simulations of a growing minimal cell. It's JCVI SYN3A, and I know most of you know SYN3A is also SYN3. When we started working on this project to do a complete model of all the reactions as well as for it to grow and divide, we were just in the process of changing it from 3 to 3A because 3.0 just had an inconsistent morphology, and I couldn't get any of my physical colleagues or biophysicists to work on the system where you don't have a cell that has a defined shape. Now we have many, many people. You see just a group of them there. The first line of people at the U of I are my students, former postdoc, a colleague, Angad Mehta, who grows the cells for people here on campus. We have people who do imaging at Harvard. Of course, we have the great group from the JCVI, their synthetic biology group. And then to do things like tomography, we have Elizabeth Vila, and I'll show you another ongoing project where we use the tomography from Julia Mohammed at the EMBL. The lipidomics was done by James Sense. And we would get nowhere with developing these 4D simulations if we didn't have the help of NVIDIA and one of my former programmers, John Stone, to check it, the models once we're done with it, because we do it at a very reduced representation, proteins or whole proteins. We don't go down to the atomistic level, but we do turn it into atomistic levels with the help of Marek and Eric Lindau using a Martini-Gromax simulations. And then for the newest treatment of the DNA, we're using models and experimental values that were obtained from Case Decker's group. So I think I mentioned everybody, so let me get started with this. So I think most of you know this SYN3A. It has only 493 genes, 452 code for proteins. And if you want to think and compare it to E. coli, just think 1/10 of E. coli, any property you could think of. Except we have many, many, many fewer proteins of unknown function. And the number keeps going down from the 90, but there's still a few essential proteins. Some of them are membrane proteins. So there's still maybe some things coming in and out of the cell that we haven't captured, but our present models of metabolism can reproduce the doubling time. So this is the one that we're going to use. And there again is a picture of the cell. It's around 400 or 500 nanometers across. And again, those are the people that are involved with the project. Hamilton, I was very fortunate to work with Hamilton Smith and Clyde Hutchinson before they sort of retired from the JCVI. Ham even studied mathematics here at the University of Illinois, but he decided when he went out to California, he wasn't going to get the Nobel Prize, or the field prize, should I say, in mathematics. So he switched into biology, and I think many of you know more so than I that he shared the Nobel Prize for the discovery of restriction enzymes. Okay, so I think that mentions everybody. And all the papers that are listed there below are papers that we wrote looking at these different components and subsystems of the system, like metabolism, the genetic information processing, changing the DNA model. That was the most recent 2023 paper. So as I said, we went about this very systematically, doing each of the subsystems. And now our whole goal is here to get a whole cell model that can run over 110 minutes of the cell cycle, which is its doubling time. And can we start getting insight into the minimal rules of life? So like any theoreticians, we got to start with an initial state. And that means just you have all this information is necessary to define that initial state. And we were very fortunate. We had the tomography in the 2019 paper, eLife paper. We had done also the proteomics plus what things are essential. We have a good estimate of some of the metabolomics, but not all of them. So we borrow a lot of information from other organisms to do that. And the transcriptomics, we've just finished up the complete analysis of that, along with publishing the proteomics for its relative SIN1. And to have all this information, as I said, we would be nowhere without experiments. That's why I always call this integration of theory and experiments, whether it's omics, super resolution imaging data, the DNA maps, all that was necessary for us to start with and get the parameters going into the model. And the parameters are going to be the reactions that you have, the diffusion parameters, so we can evolve the state of the cell in time. And we're lucky the biochemists have worked on all the enzymes involved in this organism. So we have decades of studies from them. Of course, they had to be adapted to the sequences that you have, and you had to, later on, I'll show you as we built the metabolic model, it's one thing to have a map, it's another thing to have a kinetic model for it, and you have to check that you get the free energy drops correct in this. And then you have to come up with a method to propagate these things forward in time. And since some of the components need to be treated stochastically, we have what is called hybrid stochastic deterministic methods. So like with genetic information processing, we're using stochastic algorithms. For the most of the metabolism, we're using ordinary differential equations. And then plus now to handle the DNA, we're having a Brownian dynamics description of it. And all these things have to communicate with one another. We even had to write a paper how to do that and keep the artifacts as small as possible. We do everything on the newest and best GPUs from NVIDIA, and then we can then record things like time-dependent ATP costs for any process, the mRNA costs, the DNA, state of the DNA, how much of it has been replicated, and the variation, say, things like the doubling time. So for those of you who have never seen a reaction diffusion master equation, as the name says, there are two operators, a reaction operator, a diffusion operator. And the reaction operator just looks like a chemical master equation, but all the components carry a subscript depending on where in the cell we're talking about. So you put a lattice onto the cell, and that XV can change from one lattice site to the next. And all those things, the A's in front of it are called propensities. They're related to the rate constants and to the amount of some of the substances. And you have reactions that take you out of a particular state or bring you into that state. And then you can have diffusion between subvolumes. But it's very important to take into account, is there something in that subvolume, like a ribosome, which is colored yellow here, or some of the DNA? So in everything we're showing you, everything is moving. The DNA is moving, the ribosomes are moving, the reactions are taking place. And as I said, the most recent version explaining sort of the program is in 2022 in the GitHubs that are connected to that paper and the 2023 paper. And the rest are more theoretical papers. Also, we publish things in computer science journals. So, trying to really say how we had tested the model. And as communication is a very, very important. So like just between two methods of stochastic chemical master equation and the ordinary differential equations, after a certain time and tau, that had to be determined what was the best communication time. And then also on the left, you see what got communicated. Right? And so maybe it's enzyme counts, the number of NTPs that are used in description, the deoxy NTPs to have DNA replication. And then very, very important in the system, I didn't show it in that, when I showed that first picture of the cell, failed to mention that you have to feed this thing everything. It needs to be fed fatty acids. It needs to be fed amino acids. It needs to be fed nucleosides, right? But you can recycle and you can degrade the messengers, and that keeps giving you a recycling of this. But still, they're sensitive to the uptake rates that you have. And we tested that a little bit. I think now that we have slowly a defined medium, we should do much more of that to get a really good handle on the uptake coming in through some of those transporters. That is an understudied field. And I'm even trying to get all my colleagues who are doing atomistic molecular dynamics to test that addition to us. We have some information from them that we use in the model, but there are many, many others that we would like to see tested. Okay, so is it the metabolism? You can put it down and on one slide. It was too much for any one student to do, so everybody took a subsystem. And the map itself was done in the eLife paper by a very gifted postdoc at that time, Miriam Breuer, who's now a professor in Maastricht. And then they came up with the kinetic parameters and then tested them using, machine learning and so free energy analysis. So just to give you a feel for that main glycolysis pathway, there's a phosphorelay reaction that has to take place, where as you're bringing in the glucose through the PTSG, it gets phosphorylated, and it's because it involves these three other proteins to sort of shuttle the phosphate over there to the sugar. So it finally comes in as G6P and then gets converted, and you go straight down the pathway. And that's the only place you get ATP. There's no other source of doing that. So this is very important, what you're getting made from that. At F6P, you then start branching off to either making nucleosides, or the other direction would be making lipids. Now, I always put up this picture because I want to remind people, please teach not only the simple Michaelis-Menten reaction, teach the one where you have more than one substrate and more than one product. So if you do not know the exact mechanism of binding, who binds first, who gets released first, then you have to assume a random binding model. And lucky for us, Liebermeister had led the way in trying to develop over those couple years good approximations to those. And we did it, and then we checked the free energy drop that was about 80 kilojoules per mole, and that was exactly what we predicted it should be. Okay, and as I said, always a nod, be thankful for your biochemist friends who have measured the kinetics and put the parameters into Brenda. All right, so for example, you have this PYK. It looks sort of like, good. I see what happened. PEP gets, converted to pyruvate, and it takes an ADP, and an ADP gives you an ATP. Wonderful. Guess what? That same enzyme is also used in nucleoside metabolism. So every NDP goes to an NTP using the same enzyme. Thank goodness there was a colleague years ago who measured the affinity for the various nucleotides. So the highest one is, of course, for ADP, but then they measured for the other nucleotides. And that is just one example. There's also PYK. And as I say, we would be nowhere with making a kinetic model without a lot of this kinetic parameters being made available. Now, here's our newest DNA model. We're really happy with it. It's a beads on a string, and each bead stands for 10 base pairs. And the original model, sort of based on some work that Brackley did back in 2014, we added certain features to it. Namely, we wanted the DNA to be self-avoiding, and we wanted it to avoid the ribosome, which are shown as pink here. And this has been the work of two fabulous graduate students, Benjamin Gilbert there and Andrew Mayton, and getting this thing installed and visualized, again, the help of the NVIDIA programmer, or a distinguished research engineer. I forgot his fancy title. All I know is they doubled or tripled his salary, and he left us and went to NVIDIA. And then to make and compare this model, our beads on the string model, to a more atomistic model, that's where Marek and his coworkers came into play. So in that paper in 2023, we presented how our DNA behaves and how it behaves on an atomistic level. And we're seeing if we got very similar properties. Okay, so what did we have to worry about when we deal with that DNA? DNA is pretty long, so any polymer, you have to worry about entanglement. And also, how do you ratchet it into the various positions? Well, two of the proteins that are essential, that they could not do away with, that interact with DNA, are SMCs, and they can make loops, and they can help ratchet the DNA. You get large motions of the DNA, but you can get it entangled, as you see in the middle picture. So along come the topoisomerases, they can cut it, and then you can separate the two strands. Now, we're theoreticians. We're not cutting and then rebuilding. We just soften the potential, and when you get entangled, we just allow it to pass through to one another. But again, we use the parameters for ratcheting from Decker and sort of preliminary model that was done with Micheletti, and it works quite well. And here's a picture of it. So you have an anchor and a hinge place, and then you extrude the DNA, and you have a certain distance that's determined by the SMC loops themselves. And so on the right, you're seeing what happens when we implement it. And this was the work of Andrew Maton. So I'm just showing you some of the hinges and anchors come up. The red is the origin, and then you see the yellow, it's not so clear, would become the fork, the terminus. And here we were able to replicate the entire genome in about 7 hours. It turns out it's a little faster than it really should be. So in the present cell simulations, which now take 4 days to go through the whole 110 minutes, that we're running this on its own separate GPU. So, we're giving it lots of time to sample space. All right, so let's go on. And now when we get a model, this was like before cell, is one of the preliminary forms or state of the cell. We took that, and in our paper, I think it's 2023, we converted it into this atomistic model using the Martini force field and the GROMACS molecular dynamics engine to do a complete conversion of every protein. If we didn't know its function or couldn't identify, at least we had a structure via AlphaFold 2. We've now repeated it with AlphaFold 3. And that's sort of the picture that you get. It's looking much more realistic on the right-hand side, because our membranes are just these blue, these green blocks, and you see a shadow in it. That means there's membrane proteins in it. And now at our most recent workshop that we had in May in our NSF Science and Technology Center, we built a mini version of it. Those are the instructors that were involved in the project. And then I think, yes, we even did some dynamics on it, start looking at some of the interactions. And I will tell you, even when we run these trajectories, because this cell has eliminated so much, most of the regulation, that some of the cells die. So we work with a colleague in computer science to study, put the trajectories in different categories. And so can we tell early on what cells are going to die, so we don't have to run those, because doing a full study takes, like I said, 4 days. So we're trying to assess which are the pathways, where do you have a problem? And in general, when you get to such a bare bones metabolism, something can go wrong in almost anywhere, whether it's nucleotide metabolism, lipid metabolism, or even the central metabolism. So I also want to show you then these simulations, we now can also put in the little pink guys, those are metabolites. And because we're an NSF center, we also have an official evaluator that comes and asks, what did you learn in the workshop? Are you going to use it? And, they really enjoyed this. We will have other workshops, so you are cordially invited to attend them. We only do them about once a year because they are a lot of work. But one thing we will not do is that last item. We will not do it longer than a week. It is just too much work. All right, but one of the other things that came out of then 3 months later in the summer school is we worked again with James Sense, who had done the lipidomics. He came with his postdoc from the TU at Dresden. And on the right, you're seeing, or on the left, you're seeing a representation of the membrane. And so you can see the various components. There's cholesterol, a lot of it, PC, PG, sphingomyelin, cardiolipids. They did actually the lipidomics here at our summer school in Urbana-Champaign. And then we compared the doubling time with what we got in the simulations. So the students had to do both simulations as well as wet work. They did a great job. I have to say, one of my students, who's a really great theoretician, he managed to fail pipetting. I'll say no more, but the students did a great job, right? And let's see. And here's actually some of the things that they did. So we modeled the DNA replication. We ran the kinetic model, and you can look at the protein abundances, say, after 110 minutes, had we doubled everything, at least. And then there were some outliers. And then they went over to the lab, and they did a measurement of the cholesterol, the PG, PC, and sphingomyelin, and they did it with also different fractions of fatty acids available and measured the growth curve. So, as I said, very exciting that things agreed. And there are the happy students. And at this time, we were also, every time we get a cell made, we try to put it into Minecraft, and that paper will be coming out. And so we're going to make all the Minecraft worlds available to everybody, because we did not only the minimal cell, we've looked at yeast and some cancer cells. Anywhere that we can get a 3D version of the cell, we put it in, and here's a student trying it, and I'll show you what that person is seeing. So you go into the cell, excuse the terrible music. I didn't have much choice when we did this. This movie has now gotten somewhat better. You can see very clearly the DNA. And when I told the student I didn't like the music, I said, why don't you make something like more like a roller coaster? Well, what he's done now is one of the worlds has a roller coaster in it. I want to point out, it is not in the cell. He just did it to amuse himself, to go along the DNA. And the proteins in the cytoplasm are all colored red, and the ones in the membrane are colored blue. So you get a feel of the orientation. And as you're going, you're leaving the cell and breaking through the membrane. And now, just to remind you, the kinetics making that membrane are all reactions that we're calculating. And again, those are the lipidomic studies, and that's the mole fraction. Oh, I guess I lost that on the vertical axis that that James Sense measured. And so, as I said, we are very happy with this. We can know the rate at which the lipids are being made. We also had a way to, when you're making membrane proteins, we had an estimate of what the insertion rate should be and what the ATP costs are. And then for every lipid in membrane protein, we could assign an area to it. So we could also calculate the surface area as a function of time, as well as the volume. And initially, we were sort of wondering, well, when we've doubled the volume, what should happen? And in the literature, there were several models. Is it asymmetric? Is it symmetric? What is it? But we're very fortunate, because at the same time, Tae-Kip Ha and his postdoc at Harvard were measuring or capturing some of the cells during dividing. So we were able to label the DNA, the membrane, the FTSZ, and you could catch it in the early prolate state and then the late stage, and it looked pretty symmetric to us. So in our calculations, we're using the rate at which the lipids and the membrane proteins are made, but well, I'll show you in just a moment. We just tried feeding that information directly into a Helfrich Hamiltonian that minimizes the cell surface. But instead of getting these more rounded objects, it looked like two tiers pulling apart. So later on, we get totally just guided by using the rate at which they form, but keeping to the structures that you see in the imaging. So here's what I'm talking to you about. For those of you who've never seen the Helfrich Hamiltonian, it has a long history, starting, of course, with Helfrich. The code that we're using is more from Pozeskian, and in it, there's an energy that you have to be minimizing. And those terms that are in the brackets have to do with what you're assuming for the curvature, and then the product of the curvatures. And then you can add harmonic terms for varying the surface area to volume ratio. And that's what would happen if you impose all those vertices onto our minimal cell in our lattice picture. So we kept trying to solve that as a function of time, and you'll see what happened. So over here on the right-hand side, we have what happens if you try to minimize the Hilfrey Hamiltonian. You have the SMC looping in there and the topoisomerase action. It's growing, and it's separating, but in this one run, and I'm showing you, we didn't quite get the DNA into the other side, right? And look at it. They just don't really look like the prolate shapes. So we decided, let's constrain it to the images that we see. And now this is a little boring, and the white sort of indicates the DNA. And it's up to now slowly, this double the cell volume. You're seeing ribosomes, and they're changing color here. They're yellow. They sometimes change color when they're processing a messenger. But now you saw some red flicker in there. So what we did is we labeled the beginnings of the genes so you could see the other strand being starting to form, and you'll see the white blob slowly separate. And now we're into the beginning of cell division, and it continues on. And as I said, this is what we're analyzing now. What happens to the left and to the right is that, you know, you're never going to get like 500 ribosomes left, 500 ribosomes right. It's like, you know, 510, maybe 490 over there, but that's where we are. And then you can see that here, that looks like the DNA was also almost separated too harshly, so the ribosomes moved into that area. The one thing, because the ribosomes change color from yellow to purple, we can also get the percentage of active ribosomes. That's a number that has been published in several places for other organisms. We just tend to use Dennis and Bramer as our Bible, and we get about 70 to 80%, which seems to agree. All right, so this minimal cell work was the proof of principle for us to go forward to get our STC for quantitative cell biology. But in there, we also promised to go to other bacteria, as well as try to start looking at mammalian cells and starting then with yeast cell. So this, we'd already begun to look at that through first its metabolism, already back in 2017. The example that we had of how to connect different methods was also done with the yeast on the galactose switch in 2018. So we've built that cell starting with tomography data. And here movie inside the nucleus, the gene for that transporter for galactose, which is G2, is off. And when it's on, it changes color. And then suddenly you'll start seeing one of the 100,000 ribosomes turn yellow as it's processing it. Now, those of you who know biology well know that really should be going through the ER, which we didn't have in this version. But the amount of glucose is going from 0 up to about 5 millimolar, and the number of transporters is going from about 1,000 to 15,000 within the time course of an hour. Now, we have now redone these simulations, but with putting in some ER that's become available to us from Julia Muhammad and her postdoc that just visited us. And then Angad Mehta, one of my colleagues, he already had made a photosynthetic yeast by putting in a cyanobacteria into it. Now he's trying to build in the minimal cell into yeast and see what it has to export and what the cell has to change, what has to change on the metabolism of both sides for them to accommodate each other. So that is really ongoing work as we have it. We put in the ER, we've embedded a lot of the ribosomes into the ER. We didn't have enough of the ER in the original tomograms to do a good job. That's why we left it out. But now through Julia Muhammad, we have like 600 tomograms flying from Germany over to the University of Illinois. And now I just want to say, end with just a quick word about our center. It has a very good staff. We have a research programmer who helps us. And we want to do not only models and simulations, we want to do 4D experiments. We want dynamics in our experiments. So these are some of the colleagues that are involved. I think you know many of the people that are there. Our industry partners like a barrier who make this MinFlux microscope are very important. NVIDIA, so it's not only John Stone, but other programmers from NVIDIA are helping us. And then we have the head of NCSA, Bill Gropp, and then the rest of the colleagues are from about 7 different departments, some local and some international. And this gives us really a broad range of talents to help us address these issues. And our 3 experimental techniques are MinFlux, which gives us live cell imaging at a 2 nanometer spatial resolution and a 0.1 millisecond tracking. But you still don't want to label something like a small protein. It just moves too fast. We're trying to get at the composition using label-free in-cell IR microscopy or AFM IR with a lot of machine learning. And then the tomography, we keep getting that, for our various cells. And then of course, when we try to bring the cells to life on a computer or in Minecraft, we are aided by the people who run each of those programs are members of the STC. So we also have NAMD, which was developed by my husband years ago, still here at the U of I. We have VMD for imaging. That's also a U of I product. We have GROMACS, Martini, ARBD, and of course, the one that comes from my lab, Lattice Microbes. And so this gives you an idea of the resolution that we can get to. And we have a timeline. We actually started at the very end of 2023, so we've now been around for a year, and we've already had a number of meetings and workshops. I draw your attention to the one at the very bottom. We're going to have International Physics of Living Systems meeting here from July 27 through 31. We'll be putting out an announcement. I think it may be out already. It's primarily for graduate students and postdocs to come and give short talks. And I urge everybody to apply. They want to come and give a talk on this. And this sort of breaks down sort of the thrust of our center into its various components. And then finally, let me just end by a movie or that one of the students made at this workshop in August, because they were having to do both experimental and computational work. The fellow who did this was a theoretician, and he said, well, when he thinks of the experiments, he sees something like this. And when he thinks of computations, he sees something like that. And then our idea with our center is to use experiments and build it into our computational models, but it's very important to go back the other way, because then we can make predictions which motivate more experiments. And this back and forth is really helping us to go forward, because if an experimentalist wants to know from me, what's the next thing you think you need to really measure, I have a laundry list. So with that, I'd like to thank you for your attention. And as I said, oh, that's our movie for yeast. Didn't want to show you that. That'll also be in our Minecraft paper. So be happy to answer any questions.
Thank you. Thank you so much. I really love this talk. There's a lot of questions in chat. Can you see chat?
Yep, just a second. I just have to keep an eye on the time because I have to go to FEMA with my daughter. This is terrible. So let me see. Okay, chat. Here we are.
If you can read a question out loud before answering.
Does your whole cell model include media? What nutriments come from the media? Yes, they do. Because when you make the metabolic model, and this goes back, you can see it, get a laundry list of the entire media. So it's nucleosides, it's also deoxynucleosides, blah, 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 amino acids, fatty acids. That's all in the outside media. It comes in, and for those transporters, we have a kinetic model, and we use the best approximation we can to those rates. So they are definitely there. I worked really diligently with JCVI trying to get it to a defined medium. And we almost succeeded. The one thing I'd say is semi-defined. It was at that time of the Cell 2022 paper was the delivery method for the lipids and fatty acids. They used Albumax. And if you look at the structure of it, it has multiple binding sites. And they had not necessarily, cleaned them all off and then just fed it a precise amount of, say, the sphingomyelin or the fatty acids. And then if you work with James Sense, you can start getting even better answers. And that's what he's doing now. So if you want to look at his papers, he has really studied that in detail, even down to when you have the C16s and the C18s, where are the double bonds? You just shouldn't say 18:1. You should go colon and then where is that double bond, right? That makes a difference in the overall behavior. So I hope that answered your question, Vivian. And Henry had a question, does your simulation make complex lipids or can it implement what we've learned from James Sense? Okay, so what we have in the lipid metabolism is just the formation of the two that are there naturally. It allows for an uptake rate for like sphingomyelin, and so we could adjust it, I have to say, so it would agree with the James Sense data. So we looked at what are typical uptake rates. We used those, and it agreed pretty well. So we always start with what we find in the literature, or then if we know something more from our experimental colleague, we take that. We just knew from his studies what the mole fraction should be, and that's what we tried to fit that to. And then from Aaron, oh, thank you for saying it's a great talk. Does the model have predictive power? Oh, yeah. So even in the 2022 paper, which we can only do 20 minutes of the 110 cycle, we put a lot of predictions. We tried to learn enough from those 20 minutes to set probabilistic factors, and do away with diffusion, which takes a lot of time, and put it into just a chemical master equation, stochastic ODE version of the program. Made a lot of predictions there, and some are holding up and some are not. Or we realized, diffusion plays a real role in this behavior. Also, how that DNA gets over there. We have not found an attachment organelle. We still have about at least 50 proteins. We do not know what they do, and some of them are membrane proteins, but it just doesn't look like the attachment organelle that, say, Julia Muhammad identified in the mycoplasma pneumonia. So we just put a force that's sort of been estimated has to be similar to that to help separate them. But if you push it too far or too fast, what can happen? You block the membrane, and guess what? Some of the membrane proteins can't get in there. And the degradasomes, by the way, that chew up the messenger, they are the main anchor for that. It's a complex made up of RNase Y, which is an endonuclease. And then we have two exonucleases that allows you to eat up the mRNA from either end. They have to get attached to the membrane. So when we started to look, how many degradasomes do we have left? How many do we have on the right? Oh, when too much of the DNA was on the inner surface, I think on the right-hand side, or the left-hand side, excuse me, we had fewer. It was like maybe 150 on the right and then 20 on the left. And oh my God. So we look at those. So I think these are things that once you can start doing single cell proteomics, which is still a developing field, these are all predictions and they're out there. And as I said, we use it first right now just to sort of, does this make sense to us? Because we just think it should be distributed according to the binomial distribution. You're not going to get 50-50. You're going to get a distribution around everything that you have there. Can you simulate, for example, expression of a new plasmid? Yeah, we can. Don't want to at the moment because we don't have it in there. But instead, what JCVI has very cleverly done is build in a landing pad in the Syn3A. I think the whole organism now is called Syn3B. If you want to introduce something, you can put it into the landing pad, which is very helpful. Let's see what else they got. Oop, come on, this stupid thing. This is really impressive. Why can I see all the messages? Just a second. Oh, got to get my scroll bar. Can the model algorithm be applied to the original Syn1? In principle, yes. You know what the problem is with Syn1? It has twice the genome. So genetic information processing, yeah, but it has regulation. And we have very little, if any, regulation in here. And the only thing that we would like to put in is the phosphate just builds up in our cells. Because we don't put I think it's called Phob in there, that once you have enough of it, it'll come and shut off the transporter. So you could, but then you'd also have to have more of the metabolism in there. And so somebody's got to develop that. So in principle, yes. But that's why whenever I start on a new cell, whether it's yeast or whatever, I try to come up with a minimal essential metabolism. We can make a model using that. So one would have to have the essentiality known for the Syn1. I think that might be known. Then try to make a metabolic model for it. And instead, I think what people are wanting to do now, once we publish this, I would say, if you want to check like putting in an alternate pathway, how that changes the answers, go for it. You could use this as a building model and build up complexity. Maybe, but and you could even test it by putting these things into the landing pad, but it can only hold so much. And then you're going to have to go for a longer, DNA anyhow. So yeah, there are ways to do it. Let's see. Oh, there are more questions. Yikes. Okay. Mo, could you, Emma, is that really? Yeah. Could you please speculate if model like that could ever be built for a multicellular? Oh. Yeah, why don't you do that, Emma? Yikes. We're looking at yeast, got the metabolism. We're trying to look at each organelle. The DNA scares the hell out of me there because there's just 16 chromosomes. And I'm trying to think, we've got them placed, but I'm trying to think of the simplest model we can do first. In our Cell 2022 paper, that's why we only did 20 minutes. I didn't want the cell to have grown very much. I didn't want to have to worry about DNA replication. I just wanted the genes sort of, put in lots of different configurations of DNA so we could try out, does it matter if the gene is here in this region or if it's closer to a ribosome over here. The equivalent in the yeast and mammalian cells, you have these nuclear pores. So if you do start assigning these, chromosomes, you put genes on them, even to do the galactose switch, we notice it makes a difference whether that G2 gene, which gets turned on, is it close to a nuclear pore? So it can be a difference of about 15 minutes that you see to build up these 15,000 transporters. So yes, there's so much that you would want to study before. I mean, I guess you could even go to a more coarse grain model, depending on how much, biological and chemical insight you have. People thought I was nuts to do this, or because I used to do very precise simulations. I worked with Carl Vos. We made our models on molecular dynamics models, also of the ribosome, of gene expression, the tRNA, aminoacyl tRNA synthetase interactions, its charging. I even did that reaction quantum mechanically. That took, I go, no, I'm not using that to go forward in the scale. But we know so much from the kinetics. And just beware of people developing machine learning programs that will give you kinetic parameters. You have got to check the free energy for some of those, because if your forward and backward rates don't agree with what the biochemists see, that it's almost irreversible, or yeah, you've got to test them. So I don't see any program out there. I see people trying to take the data from Brenda and develop the kinetic parameters for any organism. It's not there today, but maybe it'll be there tomorrow. The more people that work on that and know how to test it and then improve it, like I participated in the protein folding contest for years, and then I go, until David Baker came along, and I go, how come he's doing so much better on so many more than mine? And a lot of it is just using the knowledge that's out there. Look, AlphaFold 3 got the Nobel Prize, this year, and there's a reason. There's a lot of sequence data out there. There's a lot of structural data. You just have to put it together. So it's the same way. It's just, unfortunately, with most people's studies, you're not really trained very much in kinetics. And I say, until it gets to be standard that you're learning the random binding models, I don't know how you're supposed to do this. I only know the exact order in a few of the reactions, and those tend to be the ones I studied with molecular dynamics in translation, right? But there's a whole other area. And I'm just happy that they're good kinetic parameters or affinities that you can do an estimate. And then the next thing is you got to remember that you can have 2 orders of magnitude change in some of the parameters, but there are a few that are critical to get the right time separation. And we're still not far enough along that I can tell you, do this, just hold that. If we could get to that point, I think anything is possible. So I don't want to discourage you to go to multicellular. Perhaps you're as crazy as I was several years ago. And I just said, oh, there's just so much information. We just have to know how to use it and put it together. So, do what you think is best. Okay. Do you have plans, thoughts on modeling the JCI cell-free system? What new information could we get from there and that we couldn't get in the host cell models? That's a good question. I'm not familiar enough with their cell-free model. I think that with the model that we have now for the DNA replication, somebody else could try to do that. Interesting thought, because I know that's so important to build a cell. What can you just sort of take out of context and just boom? Can you just take this module and move it someplace else? I will tell you, one of the things that was really critical for our replication model is the initiation. And that has to do with how the DNA binds to the origin and what are the signatures for that and what are the concentrations, because we had some really good kinetic data that somebody had measured doing single molecule FRET. So for any size cell that you're going to look at, or if you're going to do this in solution concentrations, it has to be on the right spot, because as the cell starts expanding, then guess what? Those things can pop off. They've got to bind, twist the DNA, and then another part of the DNA has to bind to the single-stranded, and you got to form a bubble that's at least the size of the replosome. And there's a pretty good PDB structure of replisomes for bacteria in the PDB database. So yeah, you got to make a bubble that's big enough for the stuff to get in there. So you've got some cues to do, and that's what we used. And it makes a difference, those concentrations. Like if you don't have enough, you won't get it started. If you have too much, you might be able to get multiple forks. So we just got the DNA sequencing information back. And it looks like the origin to tour ratio is about 1.5 to 2, which is good. On SYN3A, on SYN1, I think it's higher, if I remember correctly. And so I think that I can completely understand that. But, what I would say is read our papers. We tried to indicate what can go wrong too. So please learn from our mistakes. So, all right, Bob, not a question. Just wanted to say, fantastic talk. Your ending made me think of this joke. Theory is when you know everything, but nothing works. Yay! Practice is when everything works, but no one knows why. In our lab, theory and practice are combined. Nothing works and nobody knows why. Oh, Bob, you are so funny. As I said, I show that all the time because the guy who was the theoretician, he is the one that did that because he said this was unique for him to come and have to do an experiment and then go and try to calculate it. And I had one postdoc who did that long ago, and uh, and it was at the charging of the tRNA. And the only thing I'll tell you is what I told him. Like normally, like when theory agrees with experiment, I always tell them, check the theory. And when the experiment agrees with the theory, I tell them, check the theory. And now I go, now you have the same person doing both. I said, just please don't lie to yourself. That's all I can ask. And I was amazed that he could get the things to agree on that. And so I think that was a paper that we put in JAX with Susan Martinez. She was head at the time of the biochemistry department. Okay, and then Leah asks, a really great work. Could you perhaps elaborate on how the lack of regulation causes cell death in your simulations? Do you think that happens also experimentally? Okay, let me answer the last part. I asked JCVI immediately, how many of your cells died? And I just got this blank stare. Like, we don't measure that. And don't start telling me, oh, there are easy ways to do it. Well, yeah, there are, but a lot of them can't be used on this organism. So it's not that simple to know. And in general, we get anywhere from 10 to 20% of the simulations will die. And so what did we see happens? A lot of it has to do, to begin with that phospho-relay system at the beginning. If you look at it, it involves PEP, but you don't get PEP being sort of regenerated till you get further down on the glycolysis pathway. So, if you're having trouble with any part of that phospho-relay, then you run out of PEP, and then guess what? Then your ATP doesn't get made, and then you just pick a place where it starts falling apart. Because almost every transporter in this system is active transport. It requires ATP. So, we did back of the envelope estimates about like, well, how much deoxynucleotides do you have to bring in to at least make, 2 copies of the DNA? How much of the nucleosides do you have to bring in to make enough of the ribosomes? So from the tomograms, you see very nicely, we got the shape, but it also told us the number of ribosomes. So do we make again 500? Yes, but then don't forget, you do have to make some messengers, right? You do get some recycling through the degradosome, but that has to balance with bringing the things in. All right, so there's that kind of regulation. And then what can happen, it's just the rates of transcription, the rates of replication, the rate of translation just all slow down. Now, in our hands, computationally, if it slows down for too long, we call that cell dead. Now, maybe in real life, it will just go dormant and then recover. Don't know. That I can't tell you because we haven't ever been able to get a good assessment of how many of the cells die. It's so hard when you have so many components, so many reactions, metabolites, enzymes running to do a good correlation plot. Because, everything moves at a different time scale. Like you double the cell at about the same time you double the DNA. Okay, but some of the other processes, they come and go, come and go, come and go. So that's why we turn to machine learning, because they could start putting them into 7 different categories. And then that allowed us, in each category, you saw there was another reaction in some of the subsystem that seemed to get into trouble, right? So for example, if you do not make enough PYK, and that's used to do this important step, of converting any NDP diphosphate into a triphosphate. You not only screw up glycolysis and getting enough ATP, you muck up the rest of the stuff too. You don't get any NTPs made at the rate that you need to keep on doing, making the ribosomal RNA and making the messengers. So each of those 7 was picking a different place. Now, when I showed this to Angad Mehta, he said, "Oh, that makes a lot of sense. When you have such a minimal cell, something can go wrong in any of them, and it'll have problems." Very, very true. But it tends to be of those 7, you understand why, right? And maybe if when you're building a cell, if you look at those results, I want to get that out. It'll be a second paper as soon as we finish this one on the dividing cell. It can help you do a plan to how to build it better. I, like, oh, maybe here's where you might need an alternate pathway, if that's, would help, relieve some of the demands on this enzyme. They're very clever, these microplasm, that they use the same enzyme to do, make 4 different nucleotides. I thought that was damn clever. But, and then as I say, James Sense has been able to make the minimal cell with just the 2 the cardiolipin and the PC that's in the organism. It just grows much more slowly. So we don't want to model that at the moment, because even doing 2 hours, it's taking us 4 days. And I'd rather get this model out, and then we can try taking away the other 2 and letting it run. We just got the newest GPUs from NVIDIA. They're these Grace hoppers. And we noticed when we put the code over there, it runs about twice as fast. So we'll think about it. But first we want to get this one out. And then I also want the data from him to check for the mole fractions. So we're trying to decide if in the next summer school we try to do that. But I go, I don't know. We got to get this thing to grow a little faster for a summer school. So that's why we just repeated exactly what we had in the Cell 2022 paper. He'll do it. He's already, I think, he has a paper either in press or ready to send out on that. So do check his work. Very interesting guy. Okay, I think I got everybody's questions, Kate.
Yes, you did. Thank you so much. That was fascinating. Basically, like you gave a second talk. Yes, I'm really glad that Mohammed asked about the cell-free system, because that's what I would ask. Is I would like to see that model. I realize the difficulties.
Well, Kate, we always wanted to try and figure out a way to collaborate. So that might be something we could think about. You have very good students, through our STC. So I have, that helps. As I think you see, to do this properly, you need really a little community, experts on each of these aspects of it. And it allows us to bring them in, like with the Marek and Eric Lindau. They're all members of the STC. And we had like a seed grant to do one of the workshops to see if it was going to be a good idea. And then his student just stayed for another month. And then we just repeated the AlphaFold 3. Because it's very important for us to be able to make good atomistic models for them to go even into the Martini GROMACS simulations. So, and I think everybody, the more that you can use to validate it, you can use molecular dynamics. I mean, look, it did get a Nobel Prize in chemistry. So even though the force fields may not be perfect, they do give you a feel if it's even reasonable, right? So yeah, we definitely can do that. And we do sort of know the problems. If the cell-free system, I just have to see what all is in it. I just don't remember, to be frank. And because it seemed to me like even from that meeting in China, they were sort of getting down replication, transcription, translation sort of in hand, right? And but just imagine if you didn't have all the tools that most cells have, like Wait, it doesn't have a PAR ABS system, the minimal cell.
Yeah.
And all the knowledge, and then, people tend to form opinions very easily because they just studied E. coli all their life. So then it would be nice if somebody would at least do a gram positive, like, bacillus maybe. Or the more often you do this on different organisms, you'll see you can get the job done. You do have to have the SMC, so that's like the cohesin, condensin, right? But even like Decker, when he did his beautiful experiments, when he measured the ratcheting rate, he used a phage DNA, but he used the cohesin from yeast, right? And
Yeah.
you would like to have it use the same system, if you're going to do it for Syn3A. So we're trying to figure out how to cut up the messengers. And that was a nice thing about the meeting in China. So he said, cut it up. If you can get the system to work on any one section of it, he'll repeat his experiments for Syn3A. And that's what I'm trying to do next. Working with on God, have we got enough of that system? It's clear you have to have ATP, but just SMC, and then I forgot what the head groups are. They have a special name. If we can do that, then, oh boy.
Yeah.
Then I'd say that would be a great help for you.
Yes.
Okay.
Absolutely.
All right. For no more questions, I think I better sign off quickly and gather up my daughter and go to the meeting.
Thank you.
FEMA is going to tell us how to fill out the forms.
Oh gosh. Thank you. I hope you wear a mask when you get to the fire zone.
Well, that's why we stayed far enough away. They're under, like, I think the one in her is Eaton Canyon. I think that's like 50% controlled. The air quality, we look at it, it's getting better, but we have masks. And they have the meeting with FEMA is just outside of the zone, right? But it's so sad. It's like the apocalypse hit here. And then you just go 50 miles further and people go, oh, we're so sorry for you, but life goes on,
I know. And people still don't believe in climate change. Anyway, thank you so much for the talk. And
all right. Bye-bye.
take care. Bye.
Will do.